Rui's lab at Sloan Kettering still continues to do great work. He's going to present this backbone panel that they developed with one of, we'll see Anna here, but our steering committee's very own, Anna Longini. So we're very excited to see this presentation. It sounds like internationally this has already been used, the backbone panel.
Yes, yes, absolutely. Thank you so much. Yeah. Thanks, Thomas, for the kind introduction. And thank you for the organizers for the invitation. It's always a pleasure. It's always a pleasure to present our work to the local New York and New Jersey community. So today I'm going to be talking about one of the tools that we've been developing for immune surveillance using a customizable mouse spectral backbone panel. Just to give you a little bit of context, the flow core at MSK has been transitioning from conventional to spectral flow cytometry. And we have a large user base, around 700 users from 250 labs. And we've been more or less developing around 25 panels a year, anything between 6 and 25 markers. So we do have a lot of demand for panel design. And so we set up a panel design service, and obviously we wanted to make sure that we could stand out compared to all the offerings that are provided by other companies, especially in two or three major points here. So we obviously wanted to make sure that any panel that we develop is the best panel as possible. So we're company agnostic. So whatever antibodies we provide, we want to make sure that they're the best, and we don't stick to just one company. The other thing that for me was very important was to make sure that we also provided some type of scientific expertise to our researchers. It happens a lot that that people just come to us and say, hey, my PI wanted me to do this panel, wanted to look at these immune subsets, but we really don't know what are the right markers to put together. And here I have to really give a shout out to Anna Longini, who did an amazing job in doing a lot of research for every request that we had, and was able to really make sure that we were building the best panel for the assays that were requested. The other important point is that usually when we're developing panels, as you probably know, there's a lot of work that goes into developing these panels and troubleshooting. And so we wanted to make sure that we would follow our researchers from the panel design to execution and even data analysis. And so we tried to put all this together and to make sure that we were really providing the best service possible. Now, very early on, we did realize that, in the literature, we have OMIBs, we have other publications that have already developed, especially here in the case for mouse immune surveillance. There's some panels out there, but one of the big issues that we had was that these are big panels and as you know, researchers will typically want to customize with different types of markers for different types of questions, biological questions that they have. And so it was very hard to, start with these OMIBs, not only because if we had to change 6, 7, 8 markers, we pretty much had to build the whole panel, and also the fact that some of these panels are not spectral. They're highly dependent on the actual instrument that was used for the particular panel. So if we didn't have that particular instrument, it would be relatively hard to not to have to develop a new panel altogether. Another thing that we also noticed is that as we were looking at developing panels, we started seeing probably something very obvious, but that just looking at these 5 random panels that we have here that we were developing, you can see that there are several markers that are represented in all of these panels. There are some markers that are only represented in 4 of them, some that are only represented in 3, but there's a huge overlap between the markers that people are typically looking at. So we really started thinking about this, and this was kind of an idea that stemmed from Mark Edinger at Cytech, that they were already thinking of a concept of a backbone panel where you could then add some drop-ins and customize your panel without having to redesign the whole panel every time you wanted to change something. So we really started thinking about it. And so imagine this, you have a backbone panel where you can look at the major immune populations, the major immune subsets. And then if you want to, for instance, look at T-cell activation, all you have to do is just put together the markers that require to go into more depth in your phenotyping and be able to characterize these populations. Or if you want to look at T-cell exhaustion or memory B cells or myeloid subsets, etc., you would be able to do this with the same panel. The only thing that you would have to do is to add the specific markers for your specific question. Another way to look at it is having this backbone panel that can characterize the major immune subsets, but for instance, looking at different tissues and maybe looking at different markers for the specific tissues. And again, you can start seeing that not only this is really interesting in terms of not having to develop a whole panel every time you want to look at different tissues or different subsets, but also the fact that in terms of data analysis, we're looking at the same panel. These are the same colors, so it also streamlines the data analysis part, which is quite exciting. So all I'm going to talk about today is culminated in this work that Anna Longini did on developing this customizable mouse backbone spectral flow cytometry panel with the help of Inij from Ross Levine's lab. And as you'll see, if you want to know more details of what we have here, feel free to look at our publication, and you'll see the amazing work that Anna did. So in summary, what is the purpose of this mouse backbone panel? So the idea is to really look at the immune subsets. And obviously, we wanted to focus on tumor samples and other disease models, since we are in a cancer center. So the idea was really to be able to characterize these major immune populations, to build a panel that we could expand or could customize without having to redesign everything all over again, with some predefined drop-in fluor spaces, and then you'll understand this better in a moment. And we wanted to make sure that it also worked for a variety of tissues, obviously, and that it would work in any spectral flow cytometer. Now, building this for a spectral flow cytometer is important because it's the only way that we can really assure that it will work in different instruments. Whereas if you had a conventional flow cytometer, it would be highly dependent on that specific instrument, and it wouldn't be as universal as we would expect. So we went ahead. So we defined, or I should say Anna defined 11 subsets, major subsets that typically our researchers look into, from B cells, CD8, CD4, Tregs, NKs, PDCs, CDCs, neutrophils, macrophages, and LY6, low and high expressing monocytes. So we have, with the backbone panel, we're able to characterize these populations, or at least that was the idea. And then you can see here that we came up with 14 markers plus a live dead that we're able to characterize all of these subpopulations. And you can see here the gating strategy that Anna built. So it's very easy to understand how we are interpreting the biology by having the gating strategy well defined for this backbone panel. Then we had to start thinking of the drop-ins. So the idea was, first, we define where we want the drop-ins to fall, and then we develop a panel with the rest of the spectral space. Now, obviously, there's some, let's say, there's some logic behind or some best practices behind building these drop-ins or drop-in spaces. So we wanted to make sure that the fluors were bright and that they were commonly available commercially. We wanted to make sure that there's minimal impact of the backbone resolution and vice versa from the drop-ins to the backbone panel. And of course, we wanted to make sure that there was minimal impact between each marker where, in the backbone panel or the drop-ins. So the first choice were obvious. So the spectral spaces in BV421, FITC or BB515, the PE and APC spaces. These are, as I mentioned, these are bright, and they're relatively easy to find any marker in one of these spaces. Now, it doesn't have to be, for instance, BV421 in particular. We're talking about the fluor spaces. So any dye that would be excited by the violet laser in this case, and that would emit in that space would be technically a good option to use in this space. Then another important point was to make sure that these were the first choice for drop-ins. And so as we built the panel, then we ended up with some spaces where we could define additional drop-ins. And here are suggestions for the additional drop-ins in the BV65, BV786, and PSI7 spaces. So putting all this together, and I came up with this backbone panel with 15 parameters, so 14 surface markers plus a live dead. And as you can see, we still have relatively a lot of space here, which we could add even more than just these 7 drop-ins that you can see here. So our first choices will always be in terms of drop-ins, will be to always look at these 4 first. And then we would look at the other drop-ins. Now, another important thing that Anna realized very soon is that, and I thought this was a really great idea. If you notice, the lymphocyte or the lymphoid and myeloid markers are actually intercalated to reduce as much as possible the interference of co-expressed markers and their respective fluors. So it ended up being a really interesting panel of 22 parameters. We don't call them colors because obviously there's a spectral flow cytometry. And so we ended up then looking at the developing the panel. And the first thing we did was to make sure that the backbone panel could clearly resolve these major immune cell subsets, or else it wouldn't be useful. And like I said before, we wanted to make sure that the backbone panel had minimal impact on the resolution of the drop-ins, and vice versa, that the drop-ins would have minimal impact on the backbone, so that the researcher or even the core facility can build these panels and just add any drop-in without having to worry about reevaluating and retesting the panel. So it's almost like a kit or a pre-made solution for our researchers. So we did all the typical, maybe I shouldn't say typical, but at least we did all of the evaluations that we could to be able to validate this panel. So things like looking at the single stain controls versus full stain to make sure that when you add all the fluors of the backbone panel, that you would still maintain the resolution and that you wouldn't add too much spread to the negative. So we did all those types of, this is just an example. There were several other things that we did to make sure that we really were able to characterize those main populations. And you can see here that this was the final result of the backbone panel, which captures very well whether it's manual or unsupervised analysis. So looking here at the t-SNE plot, and I was able to capture all of the 11 subpopulations that we were aiming to characterize, and in a very nice way. And this is done in spleen. You can see the resolution is really remarkable throughout the backbone panel markers. We did have some difficulties. I'll mention this with CD3, and we're still actually working on this. So in mouse, CD3 is not that much expressed in at least with surface staining, but you can see, and based on what Oliver was discussing this morning, that just by changing a 30-minute incubation with all the antibodies and going through a sequential staining with 30 minutes first with CD3 and then another 30 minutes with the rest of the antibodies, we were able to increase the resolution at least enough to be able to look at CD3 in all the tissues that we analyzed. Then we wanted to make sure that the backbone panel didn't impact the drop-in fluorochromes. Again, what we did here was looking at these spaces, the drop-in spaces, and we looked at unstained versus fully stained samples to see if if there was any impact. So if you see an increase in spread, this is a bad sign that you're losing resolution with the backbone panel on those fluorochrome, on those drop-in spaces. But you can see that, apart from PSI7, there is some loss of resolution, but not too much. And if you look at the other drop-ins, in fact, we still maintain pretty much the resolution that we need. And remember, these are also drop-in spaces that are that are bright. And so typically we'll have a good resolution, or at least the best resolution that we could expect. And then we went further and we made sure that adding the drop-ins wouldn't impact the backbone panel, because if you're claiming that you can add anything and then that's going to impact the backbone panel that you had in the first place, that again wouldn't work that well. And so here you can see a nice comparison. This is done in spleen cells where we added drop-ins, so 7 drop-ins in this case. And looking at the backbone markers, you can see that whether you have the backbone only or the backbone with the drop-ins, you still maintain the same resolution. And here you can see the percentages. We still capture the same percentages, whether we're adding the drop-ins or not, which is quite remarkable. We wanted to also make sure that, because here we're looking at spleen, but we want to make sure that also in other tissues, in more complex tissues, that we would still be able to use this backbone panel. So again, here you can see this is using a lung tumor, that again, the backbone panel is not impacted by the addition of the drop-ins. And again, we also captured the same percentages with manual analysis of the frequencies of a percentage of CD45 positives. I also mentioned that it was important to make sure that this worked in all of the spectral instruments. Florian mentioned this a little bit. I mean, obviously there are some nuances, there's some differences between different spectral instruments. We had these 3 in our lab, the Aurora, ID7000, and S6. And as you can see, there are some differences. If you look at certain populations here, there are going to be some differences, but we're still able to capture all of the subpopulations that we need. And again, here's looking at the percentages that we could also really identify the same percentages across the board. And the other point that I mentioned earlier is that we wanted to make sure that this was organ agnostic, that we could look at any organ. And this is really a very interesting and remarkable way to look at this. So looking at the UMAP with unsupervised analysis, you can see that we capture all the populations in all of the tissues that we analyzed, and we can do a very easy comparison. Again, the fact that we're using the same panel for all these tissues really helps in terms of data analysis, and we can quickly look at a comparison. Here's a comparison of different percentages that you see of these immune subsets in the different organs. All this with one panel. Now, again, we wanted to be sure that it also worked in tumor samples. I showed you an example of lung tumor, but here's a comparison of a pancreatic tumor sample, so PDAC versus a normal dissociated pancreas. And again, we were able to capture all of the populations without problems. Here's just a simple comparison looking at a UMAP where we overlaid both the normal and the PDAC. And you can then do whatever studies you need to do. Here's a comparison between the percentages of both the tumor and the normal pancreas, which again, you can do in a very easy way. And we were able to do this with the backbone panel. Now, one of the things that we also struggle, and that's one of the reasons why we started with a surface panel, was that we had several researchers that were interested in looking at these immune subsets with our backbone panel, but they had fluorescent reporters. And so here's an example where you have a highly expressing TD tomato population, and we were still able. So we added as a drop-in. You can see here, we had all these drop-ins, so 7 drop-ins plus TD tomato, and we were still able to capture all the subpopulations that we needed. And of course, we were also able to look at the drop-ins without any loss or significant loss of resolution, which made us very happy, of course. Now, obviously, this is what we had in our publication, so all that data is there. And I thought it was interesting just to show a couple of examples of, so we've started implementing this backbone panel with our researchers, and it's been pretty successful. Here's just one example, and I want to thank Kate for allowing us to show this data, where she had 5 drop-ins in this backbone panel. And again, as you can see, and I forgot to add here, we actually, they were also looking at BFP, And again, we have here the positive, negative populations for BFP that we could look then at the different populations that are characterized by the backbone panel. And we can also characterize all the drop-ins without impact. And you can see that by the spread of the negative, it's not increasing dramatically. And so we can still maintain good resolution for all of these drop-ins. So we're really happy, and our researchers have been really happy with this solution. And it's just streamlined the way we can provide this type of service to our researchers with very good quality. But we don't have only examples from MSK. We also have an example here. I acknowledge Mike Kistner for providing us data from Philco Puger from the Olive Lab at Columbia University. Mike has been one of the biggest proponents of the backbone panel and has been sharing even templates, helping his users to use the backbone panel. And here's a really nice example, again, able to capture exactly the same populations with the same type of resolution. I believe this was done in the ID 7000, correct? Again, with these several drop-ins, we can still, obviously, some of the drop-ins are not expressed. Obviously, it depends on the biological conditions. But again, if you look at the negative, it's really not spread. So we do have a lot of space to with good resolution. What are we doing now in terms of the backbone panel? Is the work finished? Obviously not. As you saw, we do have still some issues with the CD3 resolution. Again, in mouse, it's difficult. So we've thought of swapping the fluor. We've been using BUV 805. We have done already some work. Clara has been instrumental here. Clara Cavalcanti from our lab. She's tested some different fluors. So far, we actually haven't seen a big difference. And actually, we see exactly the same resolution for some fluors, which kind of tells us that it's not so much a problem of the fluor. It's more of a problem of expression. So far, we're not very happy with any of the changes. Now, Anna has been advocating already for quite a while that, if there's any issue with CD3, if there's a tissue where you can't really capture the CD3 population, you can alternatively use TCR alpha beta. Of course, you lose the possibility of looking at TC gamma delta, but you could add it as a drop-in if needed, if you still want to look at the different T cell populations. The other thing is we're obviously now developing a backbone panel for intracellular staining. This has a lot of advantages. Obviously, we can look at cytokines. We can look at transcription factors like FOXP3. And so we've been, so again here, Clara Cavalcanti has been coordinating this all these experiments, and we actually have already preliminary data, which looks really beautiful. We have, again, we maintain even with fixation, we maintain and permeabilization. We maintain the resolution of all the populations we were looking at. And you can see even here, FOXP3 with really nice resolution with this intracellular version. It's still not complete. We're still optimizing some of the marker of the fluorochrome choices. But we hope that very soon, and actually we plan on presenting this at Isaac, both this, but also a human version of the backbone panel. So this is going to take us, a bit more time, but we're going to start obviously with already the backbone panel that we have for mouse and just do slight modifications and see if it works as it is. We also plan to test this in more spectral systems. So we're very happy that we're going to be one of the beta testers of the A8. So we're going to test this also on the A8 very soon. We plan to test this on other spectral instruments like the Option from Agilent. And really to validate that this is working on any spectral instrument. We also have been thinking of exploring the autofluorescence extraction to improve resolution. It's not as needed if you're looking at the immune populations. We haven't seen any problems with the need to add different autofluorescent signatures. But if you're looking at, for instance, tumor markers or tissue-specific markers, that may be a problem. So we plan to build a few guidelines on how to use autofluorescence extraction if that's the case. So finally, in summary, I just want to, I hope that I convince you in a very fast way that we have developed a really nice backbone panel that's very reliable for immune profiling of the tumor microenvironment, both in hematopoietic and non-hematopoietic organs, in, like I said, in tumors and other complex immune microenvironments. We've made sure that the backbone panel maintains its resolution across different spectral flow cytometers. We still plan on testing, as I mentioned, others, but we don't expect to see major problems with that. The other thing is that we made sure that the panel was validated to incorporate 7 drop-ins. We've actually, Anna has worked already in some versions where she added around 10 drop-ins. I mean, if you have expertise, you obviously can expand it. As you saw, there's still room to add more drop-ins there. But we wanted to make sure that whatever drop-ins we recommend, that you wouldn't have to have any expertise. You can just go ahead and find those combinations of markers and fluors in those spaces, and you're ready to go. And the other thing is that we wanted to make sure that it could also accommodate bright fluorescent proteins such as TD tomato, which we showed in our publication. But we've already been seeing success with other fluorescent proteins like mCher, GFP, and BFP. So we're quite excited with that. And finally, we think we have really a really interesting, robust backbone panel that can be customized and expanded with pre-tested drop-in fluorochromes. And we believe that this really helps to save time, and at least for us now, we have like you know, it's almost like those cooking shows where you have everything ready to provide to your researchers. But it also, and I think this is very important, the fact that we have one backbone panel with specific drop-ins, it helps to bring consistency and standardization to not only to the the staining acquisition, but also in terms of data analysis, making it a very valuable solution for immuno-oncology researchers. And finally, I want to thank obviously Anna Longini. She's been the mastermind here of developing this backbone panel. An incredible work that she put together with Inês Maestre from Ross Levine's lab, who helped a lot in staining, acquiring a lot of these different versions of the panel. We want to thank also Margaret from Scott Lowe's lab. She also did some of the staining that's in our paper. And obviously, we had a lot of help. This wouldn't be possible without the support of all these companies, not only with resources like antibodies for us to test, but also with a lot of suggestions and help, which really made this successful. And I want to also acknowledge Mike Kistner. As I mentioned, he's one of our biggest proponents of implementing the backbone panel, and he's also been providing suggestions which have been very helpful for us to continue developing this. And I want to end also thanking Clara, Magda, and Gabrielle, who have been now working on the different versions of the panel. And as you saw, it's already looking pretty exciting. So I'm really looking forward to presenting this data relatively soon. And with that, thank you very much.
And we don't have any questions in the room?
Yeah, I just wanted to know if the services that you offer are they available to like biotechs or other companies outside of MSD60?
Right. I mean, so we've published this, so this is available to anyone that wants to really implement it. And actually, the nice thing of the publication is that it's somewhat of a guideline of how to implement this and even how we designed it. So we want to inspire other people to also start building these type of panels, backbone panels, that I think will really streamline the way we provide these services to our researchers.
Just asking, you know, not a biotech that comes from outside, I would like some outside help from you guys.
Yeah. Yeah, we provide external services also. So we're pretty busy with internal, but yeah.
Backbone panel that work across platforms, so I think it's very, very useful. Some people might also want to drop out some of the markers, maybe something to think about. I just wanted to address the loss in resolution and some of the drop-in detectors, if you will. The P7 did look quite significant, the loss that you have there. So just saying that, you know, it wasn't impacted too much might be, you know, a bit
An overstatement.
Put in there, right?
Correct.
Doesn't matter, but you know, looking for something rare, so that might be an issue. And obviously, if you provide the data, then people can see if they have not wanted to kind of put that. No, pay attention here. The other thing I wanted to just quickly bring up, and you know, maybe it's a matter of perspective because I was but the resolution in the S6 did not look quite as scrumptious as the other instruments. And obviously it seems like you did most of the work on the Aurora. So are you thinking of, you know, with the idea of wanting to have it be applicable across platforms, are you able to kind of optimize it on, for example, S6? You know, it basically works, but you know, it really should be better if you want to run that as a team panel.
Right.
You know, in other words, if you would have developed it, it probably would have much nicer, right? So what things, and we know that, you know, the factors are not identical, etc. So are there things that you're thinking about in terms of?
Yeah, so
Maybe still keeping that same background, but just maybe with some tweaks of, you know, incubation.
right. So that's a good question. Definitely the S6 was the one that that we didn't see the best outcome of the backbone panel. Now it's also not an easy instrument. You know, I wouldn't even say it's a fully spectral system, so to speak. So it is relatively complicated. It brings to mind all the other conventional instruments. So it's basically a conventional instrument with 30 channels, making it harder to be able to develop a universal backbone panel or a panel that would work for all the other spectral instruments and work also for the S6. So we're more I'm just going to say it, we're more thinking of the future with, I mean, even BD, their new spectral system is not PMT based, is APD based, and with more channels, and so with more capabilities, which you know, I'm sure that it will work very well for these other platforms. So we're not thinking so much on trying to optimize this panel for the S6 in specific. But there is room for improvement. If anyone wants to, you know, only has an S6 and wants to go further and like you're saying, with different staining conditions, you know, even looking at overnight staining, etc., and be able to improve somewhat the resolution there, that would be great. But we're not thinking of going in that direction. If I can.